Hello students, how are you? Good? Okay. So welcome to my next lecture, grade 9, topic cell, basic unit of the life. I hope my previous lecture is clear to you. So today we will discuss the rest part of the chapter. So nucleus. It is also known as the brain house of the cell. Why? Because it controls all the cell activity. Okay, it is dense protoplasmic body that contain hereditary information controlling cell activity as well as that is transferred to the next generation. It lies in the middle or central position. In the mature plant cell, it lies towards periphery. In prokaryotes, nuclear membrane is absent. That's why it is called nucleoid. Cells like RBC and sieve elements that lack nucleus. Cell without nucleus cannot survive for the long time. One nucleus animal is known as uninucleate but some are having more than one. These are known as multinucleate. So what is the nucleus made up of? So there are three components: nuclear envelope, nucleoplasm and nucleolus. Nuclear envelope is the double membrane covering which separate the nucleus from the cytoplasm. It contains nuclear pore. A nuclear pore allows the exchange of material between the nucleus and the cytoplasm. Now nucleoplasm. It is a colorless dense sap found inside the nucleus in which chromatin and nucleolus are suspended. Now what is nucleolus? A round structure found inside the nucleus. It is rich in the RNA. RNA is the ribonucleic acid and protein. It is the site of the ribosome formation. Ribosome is also known as the protein factory because it helps in the protein formation in the cell. So after that, chromosome. Chromosomes are thread-like structure which are formed by condensation of the chromatin during cell division are called chromosome. They referred as the hereditary vehicle which is transferred from one generation to next generation. Chromosomes are made up of the DNA and protein. A chromosomes consist of two similar threads called chromatids. Two chromatids are attached at the center by the centromere. On either side of the centromere, chromatids are present. These are known as sister chromatid or the arm. After that, chromosomes number. Chromosomes number is fixed for each species. In most organisms, they occur in the homologous pair or in the same number. In each pair, one chromosome belongs to father parent while other belongs to mother parents. Presence of two set of chromosome is called diploid. For example, 2n. 2n is that means 46 chromosome which is present in the human, and presence of single set of the chromosome is called haploid. Then 2n comes to n and 46 comes to 23. After that, role of the nucleus in a cell. Nucleus contain all the genetic information not only for the cell but also the whole organism. Its function as control center of the cell as it controls cell metabolism and cell activities also. And ribosomes are formed by the nucleolus part of the nucleus. Nucleus brings about the cell differentiation by which cell attain a specific structure and a specific function. Division of the nucleus is very essential for the cell division. Reshuffling of chromosome, that means shifting of the chromosome, or you can say change in the gene positions that will produce variations. After that, prokaryotes and eukaryotes. Prokaryotes are a group of organisms that lack a cell nucleus. They have a primitive nucleus and without bounded organelle, or you can say there is no membrane bound organelle is present. They differ from the eukaryotes which have a cell nucleus. Prokaryotes are single celled organisms which such as a bacteria and are roughly 1 micrometer in the diameter, and a eukaryotic is an organism whose cell contain complex structure inside the membrane. They having defined membrane bound structure sets eukaryotic cell apart from the prokaryotic cell in the nucleus. Okay, so prokaryotes do not have a nucleus that houses its genetic material. Prokaryote cell consists a large number of the DNA molecule compacted in an area of the cytoplasm that is known as nucleoid region. Nucleoid region is protected and encased by a cell wall or cell membrane. The outer layer of the cell that is similar to the human skin. Flagellum, it is a rubber-like device which help in the movement. So the prokaryotic cells have flagellum for the mobility. Now the difference between prokaryote and eukaryote. Prokaryotic are the small in the size 1 to 10 micrometer. In eukaryotic, large in size 5 to 100 micrometer. Prokaryotes organized nucleus is absent, and in eukaryote that nucleus is present. In prokaryote, a single DNA molecule is present suspended in the cytoplasm and nucleolus is the absent. In the eukaryotes, nucleolus is a present and several DNA molecules are also present. In prokaryotes, membrane-bound cell organelle is absent. In eukaryote, membrane-bound cell organelle is present. Typical vacuole is absent and typical vacuole is present. Mitosis is absent in prokaryote, mitosis is present in the eukaryotes. Example you can see. So this is the diagram of eukaryotic animal cell in which you can see there are different cell organelles are present, and you can see nucleus also. After that, cytoplasm. The content of the cell except for the nucleus, it include cytosol, organelle, vesicle and the cytoskeleton. Cytoplasm has two parts, cytosol and cell organelle. Cytosol is a fluid part of the cytoplasm which is homogeneous and crystallocolloidal liquid which contains substance like water, iron, enzyme, vitamin, etc. And cell organelle is the submicroscopic structure which are specialized to perform specific cellular function. Cell organelle may be bounded by a membrane as in the eukaryote, but in the prokaryotes lack membrane bound organelle. As we know that virus lacks cytoplasm as well as membrane. That's why virus is acellular, that means non-living outside the body and living inside the body. Now endoplasmic reticulum. Endoplasmic reticulum is a network of interconnecting membrane which is distributed throughout the cytoplasm. The internal compartment called the lumen is a separate part of the cell with a distinct protein and ion composition. The ER folding generates a surface area much greater than that of the plasma membrane, like the villi is present on the small intestine. At certain site, the ER membrane is continuous with the outer nuclear envelope membrane. Depending upon the presence and absence of ribosome, these are of the two type. RER, rough endoplasmic reticulum, or SER, smooth endoplasmic reticulum. RER have ribosome on its surface, whereas SER is the devoid of the ribosome on its surface. So in the picture you can see smooth endoplasmic reticulum and rough endoplasmic reticulum. In the rough endoplasmic reticulum, ribosome is present that create a rough structure. But in the smooth reticulum, there is no presence of any ribosome. Then function of the ER. It help in the protein synthesis. RER basically help in the, or it is a site of the protein synthesis. SER basically help in the lipid synthesis. In protein and lipid synthesized on ER are used for making the cell membrane. It also help in the intercellular or intracellular transport of the substance. After that, it give internal support to the cytoplasm. SCR in the liver cells take part in the detoxification of the many drugs and poison. So difference between SCR and RER: SCR ribosome absent, RER ribosome present. SCR synthesis lipid, RER synthesis protein. SCR mainly formed of vesicle and tubule, RER formed by the cisternae. Now Golgi complex. Golgi complex is an organelle of membrane-bound sacs, tubules and vesicle which secrete a complex biochemical. Golgi is a single complex in an animal cell. In the plant, they form a separate unit which is known as dictyosome. Membrane-bound sac of Golgi are called cisternae. Cisternae occur in the sacs, tube or vesicles which is present on its periphery. Now you can see the diagram of the Golgi apparatus. There is rough endoplasmic reticulum as well as smooth endoplasmic reticulum, and Golgi is present in stacks form. Now functions of the Golgi. Golgi apparatus is involved in the repair and synthesis of cell membrane. Second, lysosomes are formed by Golgi apparatus. All type of substance which are to be secreted or excreted are packed in a vesicle by the Golgi apparatus. It take part in storage, modification, packaging of various biochemical produced by different compartment of the cell. Component of the cell wall are synthesized by the Golgi apparatus. Complex and some special sugar are made by the Golgi apparatus. So these are the functions of the Golgi. I hope this lecture is clear to you. So rest part we will discuss in the next lecture. Till then, stay at home, stay safe, be happy. Thank you.